Alright, so here's your second makeup video for your January 7th lecture. So this is lecture 2A about mitosis and meiosis. So generally speaking, I'll be starting each lecture with like a easy freebie clicker question, just to encourage you guys to be here on time so you can get some easy clicker marks. So of course for these videos you won't be able to do clickers. So basically when we get to a click question, I'm just going to pause for like 5 seconds so that you can pause the video, think about what your answer would be. So read through the answers, think about what your answer would be, and just go through the problem solving and thinking. And we'll be back to recording your clicker answers on Thursday. Alright, so today we're going to talk a bit more about mitosis and meiosis. Now, you've already covered the core ideas in your pre-lecture video, but I do want to add a little more detail and depth to your understanding now that you covered the basics on your own. Just make sure that you understand it really well, because this is going to serve as a foundation for your understanding of Mendelian genetics and the problem solving that gets quite complex that you'll be doing in the coming weeks. So the way we're going to do this is we'll start with a little bit of new terminology, after which we'll go over mitosis and meiosis one more time, pointing out the differences and details between them. Alright, so first going over a bit of terminology, starting with just a tiny review of some of the key terms that we'll be building upon. So I just want you to remember that our somatic cells, so in human cells, our non-sex cells are 2n equals 46. So remember that the 46 tells us that we have 46 chromosomes in total in each of our cells, and the 2n tells us that we have 2 complete genomes. Alright, so these 2 complete genomes came from our parents. So one complete genome, so one complete set of every 23 different chromosomes came from our father, and one set came from our mother. All right. So 2n, when we have 2 complete sets of chromosomes, we call that a diploid cell. Whereas the gametes only have 1 set, we would call that a haploid cell. All right. So, with this notation in mind, you should know that when we do DNA replication and double the DNA in the cell, that doesn't actually affect the ploidy or the total chromosome number in the cell. Right? For example, this cell only has 2 chromosomes, right? So that's why there's a 2 after the equal sign. And it's also 2n, it's diploid. Okay, the cell only has 1 chromosome per genome. All right, now if we were to do DNA replication, so that these unreplicated homologous chromosomes now become kind of replicated chromosomes, so chromosomes that have 2 DNA strands, 2 sister chromatids per chromosome, right? This is still a 2n equals 2 cell. It's still a diploid cell with 2 chromosomes, because when we're counting the number of chromosomes for ploidy, or for number of chromosomes, as long as these 2 sister chromatids are still stuck together, this counts as 1 chromosome, second chromosome here. So this is a diploid cell with 2 chromosomes. This is also a diploid cell with 2 chromosomes. The only difference is this one here is unreplicated, this one here is replicated, right? So keep those in mind. All right. So first clicker question is, what do these homologous chromosomes share between them? Okay, so if you're looking at a pair of homologous chromosomes, right? So pause it now, think of your answer. Okay, and I'm assuming you chose your answer. Correct answer here is B. All right, homologous chromosomes have the same genes, right? They have a gene for hair color here, and so forth, but not necessarily the same alleles, because the copy of the hair color allele you got from your mom might be for blonde hair, the one from your dad might be for dark colored hair. All right, so the same genes coding for the same proteins, but the alleles, the versions of the proteins that they code for, can be different between homologous chromosomes. Second question, so pause now. Okay, and continuing on, so sister chromatids, for sister chromatids, answers A and D are both true. All right, so they share the same genes and the same alleles, as well as the exact same DNA sequences, unless there's any mutations, because you did DNA replication to take this one DNA molecule and make 2 exact copies of it. All right, so it makes sense that both the genes and the alleles would be the same for sister chromatids, even though the alleles could be different between homologous chromosomes. All right, so getting into some new terminology then. All right, so equational cell division, right, is when the cell divides in such a way that both the parent and the daughter cell, so the cell we have at the beginning and the 2 cells we have at the end, have the same ploidy. All right, meaning they have the same number of genomes. All right, whereas a reductive cell division is cell division where the daughter cells have only half of the ploidy of the parent cell. All right, so to apply that to what you know about mitosis and meiosis, here's the first question. All right, so pause it now. Okay, and now that we're back. So mitosis is equational because before and after mitosis, the parent cell and the two daughter cells have the exact same number of chromosomes. All right, so it's equational, right? Why does mitosis have to be equational? All right. Oops, gave it away there. So mitosis has to be equational because if you think about it, you're using, when you grew up from a single cell to your adult bodies right now, that cell had to divide again and again in order to grow up to your adult bodies and to maintain them as well. And as that cell is dividing, we want to keep all of the DNA, we want to keep all that information the same from cell to cell. All right? So we definitely don't want to be cutting the amount of DNA we have in half as we're growing our bodies. All right? Now for meiosis as a whole, what kind of cell division do we have? So pause it now. Okay, so hopefully you chose reductive. All right, so meiosis is in general reductive because the gametes you produce have half of the chromosomes that the original parent cell had, right? So why does meiosis actually have to be reductive? Why can't it be equational? Okay, so pause it now. Okay, so for diploid organisms like us, we have to have reductive meiosis when we're making gametes because we need to use meiosis to make haploid gametes that have only 23 chromosomes, so gametes being the general term for sperm or for eggs. This way, when we fuse a sperm and an egg together to make a new diploid cell, right, it would only have 46 chromosomes again. All right, so if we didn't reduce the number of chromosomes in the gametes by half first, then every time that we make gametes, we'd be making gametes with the full set of chromosomes, 46 of them. And when we put two gametes together, we would get an offspring that has twice as much DNA as we do. That would keep doubling every generation, so that wouldn't really make sense. All right. So moving on, let's take a look at the slightly bigger picture than just mitosis and meiosis. All right. Now, so before we get into mitosis and meiosis, the details of that, let's put them in the context of the cell cycle as a whole. All right. So cell division, right, which includes mitosis or meiosis, which is how we're dividing up the DNA, coupled with cytokinesis, which is how we divide the rest of the cell. Okay, that's how those two things are separated. So mitosis or meiosis plus cytokinesis together will produce cell division, will cause the cell to divide. Right? Now this is actually just a very short period of the entire cell cycle. Right? A very short, small part of the cell's everyday life. For example, even if you look at the onion root slide that you might have seen in 101, where these cells are actively dividing to grow the onion's roots as quickly as possible into the earth, notice that most of the cells still have kind of normal-looking nuclei. They're not really dividing. They're all in this interphase here. And it's only these ones with these kind of spaghetti-looking chromosomes that are in M phase. Right? So notice very few even in an actively dividing tissue. All right? So cell division is a very small part of the cell cycle. Okay, so we call this cell division phase the M phase, whereas everything else is the interphase. Okay, so the rest of the cell's life cycle is the interphase. So everything from one cell division until the cell's ready to divide a second time, that's all interphase. Okay, inter meaning in between M phases. All right. So what happens during interphase? Well, the main thing that cells do is it actually does whatever job that cell is supposed to do. All right, because during M phase, right, while the cell is kind of getting the DNA ready to divide and go into two separate cells, the DNA gets really tightly coiled. If you think about spaghetti coiling on a fork and then a whole bunch of spaghetti forks coiling into another kind of bigger rope of spaghetti fork things, right? That's kind of how the chromosomes are being wound up by the histone proteins into these tightly wound small structures here, right? So during the M phase, the DNA is so tightly coiled that you can't actually access the DNA to express the genes and make proteins. All right, so it's only during interphase when the chromosomes are kind of fully loosened up. So you see that this tightly wound chromosome is actually spread out across this wide area here, just like the spaghetti is kind of loosely spread out here as well, right? So when it's relaxed into this massive kind of unwound chromatin, that's when gene expression can actually happen. So during most of the cell's life cycle, it's unwound, the DNA is unwound, and you can express all the proteins, all the genes that you need, right? Now some cells actually just stay as these kind of actively working cells, and they don't actually divide, right? So that's a cell that's in the G0 phase. Okay, so it just kind of stays in this area here and just stays alive and keeps doing its job, right? For example, heart muscle cells don't tend to divide once you're a full-sized adult, right? And so basically, instead of continuing on with interphase and preparing to divide, it basically just stays alive and contracts and keeps you alive. So if the cell is not going to be in G0 and it's going to divide soon, it's going to have to go through the rest of interphase and go towards M phase. All right. So for actively dividing cells, we first go through a G1 phase, right, in which the cell's growing and it's making more cytoplasm. So making more organelles, more ER, more mitochondria, all that kind of stuff, in order to make the cell bigger for it to divide. During the S phase, or the synthesis phase, you get DNA replication, right? So way before you start dividing the cell, you've already doubled the DNA. All right. So this is where your chromosomes go from unreplicated chromosomes with a single DNA strand to replicated chromosomes with 2 DNA strands, 2 sister chromatids each. All right. So that's already in existence at the end of this S phase. But remember, you don't actually, as much as we like drawing them like this, you don't actually see these chromosomes yet. There's still this loose kind of chromatin, okay, during S phase. And then during G2 phase, it's another growth phase where the cell continues to grow and makes more cytoplasm. There's also checks and balances there to make sure the DNA was replicated properly before it grows and divides further. All right. Okay, so that's what happens during interphase. So by the end of interphase, we have doubled the DNA already. The cell's nice and big. It's got plenty of cytoplasm to go around, and it's ready to divide. All right. So next, we're going to go through mitosis in detail. So having learned what happens to the chromosomes in your pre-lecture video, these details should be pretty easy to understand. That was kind of the point of having you watch the pre-lecture video first. So what I'll do is I'll teach this to you once first, and then I'll ask you a few clicker questions to really kind of dig deep and see if you understand it in a functional, kind of flexible way. All right. Along the way, I'm going to highlight some things in red just so you notice them more and notice how they're going to be different from meiosis when we go over meiosis next. Okay. So note though that even though the figures that show the phases of mitosis always show interphase at the beginning, interphase is not part of mitosis. Okay, they just show a picture of interphase there, just to allow you to compare what it looks like in interphase before mitosis starts. Okay, so interphase, this is what a cell looks like before it starts dividing. DNA's kind of relaxed, chromatin, nuclear membranes intact, right? When we transition to the early mitotic prophase, as we start doing mitosis, right, you notice that the chromosomes are starting to coil up and become visible. All right, so no longer loose chromatin. We're starting to see these little noodles here. So again, imagine this loose chromatin being coiled up into kind of more densely packed visible chromosomes. All right. So by the later parts of mitotic prophase, you'll find that the nuclear envelope's gone. So you notice the DNA is kind of more spread out within the cell because with the nuclear envelope gone, there's more room to move the chromosomes around. All right. At the same time, the mitotic spindle is forming out of these microtubules being synthesized, and these spindle fibers will attach to both sides of each chromosome's centromere. So notice here's a chromosome. This is the centromere, the skinny part there. There's these kinetochore proteins on either side, right, that attach to these microtubules, one on either side of each chromosome. All right. So by metaphase, the mitotic spindle is fully formed, right? The mitotic spindle being this kind of structure of microtubules here. And as those microtubules pull on either side of each chromosome, it ends up pulling the chromosomes in line across the middle of the cell, across the metaphase plate. All right. And notice that each of these chromosomes are lined up individually. That's highlighted in red because that's different to meiosis. All right, so now we're getting ready to separate by the end of mitotic metaphase. Now, in anaphase, each pair of sister chromatids, right, so the 2 sister chromatids of this light blue chromosome, for example, will separate so that one goes up, one goes down. All right. With that separation, each of these now new chromosomes are separated into either pole of the cells because they're being pulled by these kinetochore microtubules. So kinetochore microtubules are the microtubules that are attached to the kinetochore at the centromere. Okay. Now, non-kinetochore microtubules at the same time, so these microtubules here that aren't attached to chromosomes, they interact with each other and push each other apart. All right, and that causes the cell to get longer, get elongated along the line that joins the 2 poles, and that actually helps to kind of get the chromosomes even further apart. So these kinetochore microtubules pull the chromosomes up and pull the chromosomes down. The non-kinetochore ones push each other away to make the poles separate, to elongate the cell. All right, that's what happens during anaphase. So finally, telophase and cytokinesis kind of happen kind of overlappingly together. So in mitotic telophase, the chromosomes start to uncoil, right? The nuclear envelope reforms. Essentially, everything that happened in prophase happens backwards in telophase. All right. The spindle also becomes dispersed as well. All right? So essentially, it's kind of turning back into a normal cell that's not dividing anymore. while the DNA is uncoiling, being repackaged in the nuclear membrane, is uncoiling, being repackaged in the nuclear membrane, and the spindle is going away, cytokinesis happens. So cytokinesis is the separation of the cell into two daughter cells. All right. So for plants, that happens by the building of a cell plate, where kind of vesicles come together with cell wall and cell membrane material, and they build a new wall to separate the two cells. Or for animals, you can get a cleavage furrow, which is when kind of the middle of these cells pinch in together more and more until they pinch each other apart. Okay. So that's pretty much mitosis in a nutshell. All right. Feel free to review that again in the video if you like before going on to meiosis, just to make sure you understand it well. All right. So to help you kind of digest what you just learned, let's go through a few clicker questions. Again, I will tell you when to pause it, and then just wait a few seconds, and then I'll continue on, assuming that you paused the video to really think about what the answer should be. Okay, so here is your first clicker question. Please pause it to read it and to think of your answer. So pause now. Okay, so coming back, so the answer here is B. All right, so an organism that is 2n equals 4, even during mitotic metaphase, where all of the chromosomes are obviously replicated, and there's actually 8 DNA strands here, there are still only 4 chromosomes. 1, 2, 3, 4. Okay. Second question, think about it, and pause it and think about it now. Okay, and we're back. So I kind of gave it away a little bit in my explanation of the previous question, and so there are 8 double helices, right? So 2 sister chromosomes, so 2 DNA molecules here, 2 here, 2 here, 2 here, for 8 total. All right, another question here. Please pause now. Okay, and coming back. So during mitotic anaphase, this is kind of a weird phase where it's kind of in between, but there are 8 chromosomes. Remember I said that as long as these two sister chromatids are stuck together, this is only 1 chromosome. So as soon as they're separated, these are 2 separate chromosomes. So this is kind of like a weird phase here where there's actually 8 chromosomes in the cell. Okay, we don't bother counting ploidy in this cell because this is a very short phase. Okay, last question for this section. Pause now. Okay, and coming back. So the answer to this one is 4. Right? So after mitosis and cytokinesis is done, since we started with 4 chromosomes in the beginning, and this is mitosis, it's an equational cell division, we should see 4 chromosomes at the end as well. All right? So we haven't changed the number of chromosomes at all. All right. Oh yeah, and sorry, there's one more question at the end here, so please pause now and think about it. Okay, and we're back, and hopefully you chose no, right? So we started off with replicated chromosomes at the beginning that are ready to divide. Once you've divided them up, each cell is just going to have 1, 2, 3, 4 chromosomes that are unreplicated. It's just a 1 DNA molecule each. Okay? So, moving on to meiosis. So let's start with meiosis 1. Okay? And while we go through meiosis 1, we're going to take our time and compare it with mitosis at the same time. So I'm going to do meiosis 1 stuff in blue and mitosis in green and so forth. Okay? So as I go through this, I'm going to have the pictures for meiosis up top, and then the same figure that you saw earlier for mitosis underneath, so you can basically line them up and think about them. Okay. Along the way, notice the similarities, which will be in black text, and also the differences, which I'll highlight in red text, so you can see what are the actual main differences between meiosis 1 and mitosis. Okay. Please note that we're keeping kind of the same type of cell for both examples, so the original parent cell for both mitosis and meiosis are going to be 2n equals 4. So a diploid cell with 4 chromosomes only. Okay, 2 long ones, 2 short ones, 2 blue ones that came from dad, 2 red ones that came from mom. Okay, just like in your videos. Alright, so let's start with prophase 1. Okay, we use the Roman numeral 1 because we're looking at meiosis 1, and all the other phases are named the same way, prophase 1. So in prophase 1 of meiosis, the chromosomes basically coil up, the nuclear envelope breaks down, all this is the same as mitosis. And as the chromosomes are condensing, though, here's something that's new. The chromosomes are actually synapsing with its homologue. So this chromosome and this chromosome are a homologous pair, right? And they're synapsing, they're sticking together, okay, forming close associations in order to form a tetrad. So notice that a tetrad is this structure here. 2 homologous replicated chromosomes stuck together, so there's 4 DNA molecules stuck together. Okay, so once they've been synapsed, you can also notice some chiasmata, right? Meaning these kind of cross-shaped structures here. which kind of tell us, under a microscope, we can actually see where crossing over is happening between these chromosomes. So as you saw in the pre-lecture video, when there's crossing over happening, you're actually breaking both strands at the same point and swapping the pieces so that you get different combinations of alleles from the parent, from the two different parental chromosomes mixing together. All right. Notice another difference is that in prophase, just for mitosis, the spindle fiber is attached to either side of each chromosome, right? Whereas in meiosis 1, the spindle fiber is going to attach one to this red chromosome here, just on one side, and one to this blue chromosome here on the other side, okay? Not two to every chromosome, all right? So each tetrad gets one spindle fiber on one side, one on the other. Okay, so that in this way, each chromosome's centromere only has one spindle fiber attached, and that's very important. You'll see why in anaphase 1. Okay, so in metaphase 1, you can see the spindle better. You can see the differences between mitosis and meiosis 1 better. All right, so in my metaphase 1, when the spindle's fully formed and everything's lined up properly, notice that in meiosis 1, the chromosomes are lined up such that the tetrad is stuck together with one spindle fiber on this side, one on the other, one on this side, on the other, instead of all four lined up individually. Okay, so each chromosome lines up with this homolog across the metaphase plate. All right. So in anaphase, you also see the differences quite clearly in that as things separate, we are not separating the sister chromatids yet. Right? We're actually pulling one of the homologous pairs, so one whole chromosome up, one whole chromosome down. All right. So each replicated homolog with both sister chromatids still attached one will go up, one will go down. All right. So once again, notice the difference: one full chromosome going up, whereas four kind of separated, new chromosomes separating. All right. Other parts are still the same. So non-kinetical microtubules push apart, elongates the cell, helps with separating the chromosomes and the cell itself later. All right, telophases and cytokinesis is pretty much the same. So the chromosomes will uncoil, nuclear envelope reforms, spindle is dispersed, and then cytokinesis separates the parent cell into two separate cells. So all of that's the same as mitosis. All right, some questions to help you digest that. Okay, so these questions are similar to before, but now we're asking about meiosis 1. All right, so here's the first question. Pause and think about it. Okay, so coming back. Answer to the question is B, 4. All right. So during metaphase 1, again, we have 1, 2, 3, 4 chromosomes. Okay. Next question. Pause it now. Okay, and coming back. So in anaphase 1, we still have 4 chromosomes. So unlike anaphase in mitosis, where we now have 8 chromosomes here, we still have 4. 1, 2, 3, 4. We haven't separated sister chromatids, so this is still just one chromosome. All right. Next question. Pause it now. And we're back. So answer here is 2. Right. So how many chromosomes are there in the daughter cell right after meiosis 1 and cytokinesis? Well, look here. Right. We have 1 chromosome, 2 chromosomes. Right. 1 chromosome, 2 chromosomes. Right. So essentially, we've already cut the number of chromosomes from 4 down to 2. Yes, they're still replicated. Yes, there's still kind of two DNA strands here, 2 DNA strands here, but we count 2 chromosomes only. All right, so these haploid daughter cells have only 2 replicated chromosomes each, and when we're counting ploidy, it's a haploid cell with 2 chromosomes now. All right. So after telophase 1 is done, so between meiosis 1 and meiosis 2, there is a very brief interphase, right? We call it interkinesis because it's kind of in between meiosis 1 and meiosis 2. It's kind of in between phases here. Okay, so during this interphase, we don't do DNA replication. That's very important because we don't want to double the DNA. We can't anyways because notice the DNA is already doubled. It's already replicated. We can't do DNA synthesis again. All right, so this is great. We have a haploid cell. All we got to do is separate these so we don't have replicated chromosomes anymore. All right, so each cell is already haploid, right? The chromosomes are still replicated, so we just have to separate the sister chromatids with meiosis 2. Okay, so for meiosis 2, everything's pretty much the same as mitosis, except that we only have 2 chromosomes, whereas mitosis in the same animal would have 4. Okay, so in prophase 2, the chromosomes coil up to start to become visible. Nuclear envelope breaks down, spindle fibers start to form, and they attach to both sides of each chromosome, just like they do for mitosis. Okay, notice we're not forming any tetrads this time because they don't have homologs to synapse with, right? This short chromosome doesn't have a second short chromosome in the same cell for it to synapse with, so it's by itself. Okay, so pretty much same as mitosis, except we're looking at 2 chromosomes. Okay, for metaphase 2, notice the mitotic spindle is fully formed, and now we see each chromosome lined up individually across the metaphase plate. Okay, there's only 2 chromosomes, but it looks the same as mitosis. In anaphase 2, now we're finally separating the sister chromatids, okay, for each of these chromosomes. And they move along the kinetochore microtubules. The non-kinetochore ones push things apart in order to elongate the cell. So pretty much the same as mitosis, except only 2 chromosomes. Finally, telokinesis and cytokinesis, same thing. Chromosomes uncoil, nuclear envelope reforms, spindle goes away, and cytokinesis separates the cells. So each of these 2 cells becomes its own 2 more cells, so 4 in total. Okay, so moving on to a few more questions now. So here's one question. Please pause to read it and think about your answer now. Okay, coming back now. So a triploid cell, a cell that has an odd number of genomes, right, can do mitosis. Mitosis doesn't care how many homologous chromosomes there are because each one just acts individually. Each one lines up and splits individually. But for meiosis, it can't consistently do that properly because if you have 3 homologous chromosomes, 2 of them are randomly going to pair together. The third one just doesn't really know where to go. Randomly, it'll kind of go right for one cell so that there's 2 green ones here, and then not for the other side, so there's only 1. Okay, we'll talk a bit more about that in a later lecture. Another question here, please pause now. Okay, now we're back. This is just to review the terminology. So mitosis is equational. Meiosis 1 is reductive because it's meiosis 1 that we go from 4 chromosomes down to 2 chromosomes, from diploid to haploid. Therefore, this must be the reductive cell division. But then from here to here, for meiosis 2, we're going from 2 chromosomes that are replicated to 2 chromosomes that are not replicated. Right? So that's an equational division. Okay, so hopefully that solidifies that terminology. All right. In terms of ploidy, right? Notice that we are 2n equals 4, 2n equals 4. Mitosis keeps everything the same. We went from 2n equals 4 to n equals 2, so haploid, to n equals 2, still haploid here. Okay, so if it helps, you can note down the numbers to keep track of the ploidy. Okay. Okay, I guess that's about it. So thank you for listening. I will post your next video as soon as I can.